Bismillah ar-Rahman ar-Rahim. 5090 syllabus 23 onwards, O-level biology. This is the second video on chapter 8. I took a little bit of a time in between, didn't upload any videos, but now I will continue regularly inshallah. So this is the video 2 of the chapter 8. In this video we should continue with the 9. Describe the functions of the main regions of the digestive system. Describe the functions of the main regions of the digestive system. So we've got to discuss all these functions right from A, B, C, D here till here. Then describe the functions of amylase, maltase, protease, and lipase. Then describe the function of the HCl in the stomach. Then understand that different proteases present in the stomach and the duodenum work best at different pH levels. Then outline the role of the bile, and then describe peristalsis. So this video is going to be containing this part of the syllabus. First of all, let's get an overview of the different parts of the alimentary canal. So you've got the mouth, which has the teeth and which has the tongue. And then you've got the 3 pairs of salivary glands, 3 on this side, 3 on that side. Then you've got the esophagus. There is an O with it, O-E-S-O, but you don't pronounce the O. So esophagus. Then we've got the stomach. The esophagus, this is the esophagus which leads into the stomach, and there is a sphincter here and there's a sphincter here, which is going to control the input into and out of the stomach. Then we have the liver, and then we have a gallbladder which stores the bile. So the liver produces the bile and the gallbladder stores the bile. Then we have the pancreas. Then we have the first part of the small intestine, which is called the duodenum. And then of course it continues into all this here. All this is the small intestine. And then of course, starting from here onwards, this is the large intestine. And then you have the rectum and the anus, the anal sphincter through which the feces pass out of the body. So first you must know the labeling, but then the most important thing is the functions, which is important. Now, what are the functions of the mouth? The mouth functions are, number 1 is ingestion. Ingestion means taking in food. You put a piece of cake in your mouth, you put a piece of apple in your mouth, you take some French fries and you put them in your mouth. That's ingestion. Then what is going to happen is, in the mouth we have the teeth. The teeth are a very good tool for physical digestion. Now you cannot say chewing. Why can you not say chewing? Because you have a chewing gum the whole day in your mouth and nothing happens to it. It remains the same size. But you've been chewing it the whole day. So please do not use the word chewing. That's wrong. What do the teeth do? They do grinding and crushing. And this is physical digestion because it just breaks up the food, whatever you've had. If you had a piece of apple, well, it just breaks up into tiny, tiny pieces. If you had fries, well, you're going to have, it's going to just break it up into tiny pieces. And then it's going to mix with the saliva. And with the help of the tongue, it's going to sort of move around in the mouth. And then you're going to be able to swallow it so that it enters your esophagus. But here, we also have the 3 pairs of salivary glands. The 3 pairs of salivary glands, this one here, and this one here. They produce a fluid called saliva. And that contains an enzyme called amylase. And the amylase breaks the starch into maltose. Now this is a molecule of starch, and you see how I'm breaking it up. Every second bond is broken. So 2 glucose units, this is 1 glucose unit and this is another glucose unit. Now these 2 glucose units together, so we have 1 glucose and then we have another glucose unit joined together, and this is called a maltose molecule. So it is going to break up every second bond. And this is going to result in starch to maltose. So basically the mouth has, number 1, ingestion, number 2, physical digestion, and then number 3, chemical digestion. Now coming to the role of the stomach, where is the stomach found? Now this is the stomach, which everybody can see here. This is the stomach. So this is the esophagus ends here, and then the duodenum starts here. So this is the stomach. This whole thing is the stomach. Now what we need to understand is that in the stomach what happens is a hollow organ. It's like a sac. First of all, there's going to be physical digestion by the churning of the food, because the muscles of the wall of the stomach press on the food and they churn it. It's just like mixing them up. Like you put banana and milk in the machine and you just churn it, and that whole thing becomes very, very homogeneous, and it becomes the pieces of mango or banana just broken up to tiny pieces and mixed with the milk. So you're making mango milkshake or banana milkshake. So physical digestion, churning of the food. Chemical digestion, there's an enzyme which is produced by the cells in the wall of the stomach, and that is a protease enzyme which digests the protein to peptides. And then, of course, we have a famous acid, HCl, which destroys pathogens, because when you eat, the food might contain a lot of pathogens because they have come from the air or with your hands. So it destroys the pathogen and it provides the correct pH for the enzyme protease to work. And this is part of the gastric secretions. So the gastric secretions are made up of HCl and the enzyme protease, which we also call pepsin. But as the syllabus now has slightly deviated, so it's just being mentioned as a protease and the presence of hydrochloric acid. Now the food remains in the stomach. There's a sphincter which closes it here, and there's a sphincter which closes it here. A sphincter means a ring of muscle which is going to keep it closed. So the food stays here in the stomach for about 2 to 3 hours once you've had your meal. And it stays there so that it's mixed with the acid, and if there's any protein in it, that is going to be digested. Please remember, no carbohydrate is digested and no starch is digested, no carbohydrates, no fats are digested. And the amylase which was coming from the salivary glands, the saliva contained the amylase, that will also not work here anymore because the pH is too low for it to work. Now coming to the heading of small intestine, the small intestine is made of the duodenum. The duodenum is the first part of the small intestine, and then it continues into all this. So this will be the ileum. So duodenum and ileum will be the small intestine, and in this we're only going to talk about chemical digestion. And we have 4 enzymes here we're going to talk about. Number 1, amylase, number 2, maltase, number 3, protease, and number 4, lipase. Now all these enzymes are going to work in the small intestine. The amylase comes from the pancreas. The maltase is produced in the small intestine, the cells of the small intestine. The protease also comes from the pancreas, and the lipase also comes from the pancreas. So we can see that the pancreas, so in the pancreatic juice we have the amylase, the protease, and the lipase, but the maltase is present in the cells lining the small intestine. Now let's look at the action of starch to maltose. So there's an enzyme called amylase, and then you can see how I've showed you it breaks every second bond. Now the maltase enzyme will break every individual bond. So now you have glucose. Now this glucose is enters the blood. Now it can enter the blood and will be carried around the body, and then of course this glucose will be respired to release energy. We come to the function of proteases. Now the 2 proteases which are mentioned in your syllabus are pepsin and trypsin. Now pepsin is found in the stomach. Trypsin is produced by the pancreas and is found in the small intestine. So this is what we need to understand is present in the small intestine. Now the proteins will be converted to amino acids by pepsin and trypsin. Both of them are a protease. But actually it happens in 2 stages. First the proteins are converted into peptides, and then the peptides are converted into amino acids. So this has to be clear to you all that it has to be in 2 stages. But though this is not mentioned in the syllabus, but I like to tell you about this. So they just say proteases convert protein to amino acids. The pepsin is produced in the stomach, the trypsin is produced by the pancreas, but it works in the small intestine because the pancreatic duct opens in the duodenum, and that brings the pancreatic juices which contain the pancreatic amylase, the trypsin, and the pancreatic lipase. Now, if you look at a molecule of fat, it's also called a triglyceride. You know, understand the reason why it's called a triglyceride, because it is made up of one glycerol. I said it is made up of one glycerol molecule, which is 3 carbon, and it has 3 fatty acid chains. So what does the lipase do? It breaks this bond, and these are ester bonds. We study a lot of this in A levels. So the fat, as we say, the lipase, what does the lipase do? The lipase breaks up the fat into fatty acids and glycerol. The lipase breaks up the fat into fatty acids and 1 glycerol. So the lipase is also produced by the pancreas. And this also works in the, of course, it's poured into the duodenum, but we say it works in the small intestine. And this is when the fat are going to enter the lacteals, and then they're going to be carried in the lymph, and then they will finally reach the blood and they'll be used by the body. Now here is a diagram in which I want you to see the liver. And the liver is a huge organ which is placed here, you can see on the right side of the person. And there is a gallbladder. So the liver produces bile. I said the liver produces bile, but the gallbladder just stores the bile. So the liver produces bile, so that means it is produced in the cells of the liver, and then it is carried and stored in the gallbladder. And then there is a bile duct which is going to open here in the duodenum. This is the duodenum. And in the duodenum also opens another duct which is called the pancreatic duct. This is the pancreatic duct. And you see both these ducts join and they open here in the duodenum. So there's one opening. The pancreatic duct and the bile duct join and pour their secretions. And both contain alkalis. The pancreas also contains an alkali and the bile also contains an alkali. So both of them contain an alkali, and this of course is very important because it neutralizes the acid which is coming from the stomach, because the food is mixed with the HCl, and that is of course going to destroy the lining of the duodenum. So we don't want that to happen. So all that acid is neutralized by the alkali. You can see another diagram. I like this diagram very much. You see the liver, I said, produces bile. And then I said the gallbladder stores the bile. So it's just a store. And some people develop stones in the gallbladder. I know I have a lot of stones in my gallbladder. But they don't do me any harm, so they're there. So these salts deposit out and these stones are formed in the gallbladder. And then you can see they're opening in the duodenum. And you can see here the pancreatic duct is also coming and the bile duct is also coming. And it's opening here, which is called the ampulla of Vater. But you have to understand is, of course, these are some details which are not required by the O levels, but I just like to mention this. So the liver is where the bile is produced, the gallbladder stores the bile, the pancreas produces an alkaline solution which is containing all the three enzymes, the pancreatic amylase, the pancreatic protease, which is trypsin, and the pancreatic lipase. So the pancreas is producing all these. And this is going to help us in the digestion of the food which is entering the duodenum. Now coming to the next heading is ileum and colon. Now basically their function is only absorption. In the ileum, all the digested food is going to be absorbed. All that is absorbed in the ileum. Nothing enters the colon. In the colon, just a lot of water is absorbed because the gastric juices is a lot of water which is thrown into the intestines. So all that is absorbed in the colon, which is the large intestine. But the absorption of water, glucose, amino acids, fatty acids and glycerol, vitamins and mineral salts. You had oranges, vitamin C. You had milk, calcium. You had vitamin D in dairy products. So if you had a lot of meat, well, you had iron in it, and that iron is absorbed as well. So all this will be absorbed in the ileum, and mostly in the ileum. Colon just absorbs a lot of the water which is left from the gastric juices. That will all be absorbed into the blood and will be, of course, part of the will keep the volume of blood constant. Now, the last part which we must know is the rectum and the anus. The rectum is where the stored feces are stored for the short while that you don't defecate. And the anus contains the anal sphincter, which relaxes and allows the feces to be passed out of the body. So basically, that is called egestion. Just like you had ingestion, taking of food, egestion is the removal of this undigested food. which we cannot digest, like cellulose we cannot digest, lignin we cannot digest. So all that food which we ate, which contained all this material, which we don't have the enzymes to digest them, so that has to be then passed out in the form of feces out of the body. And that would be the part of the rectum and the anus which is responsible for this egestion. Now, another factor to understand is that the pH, the optimum pH of the enzymes are very, very different. Now, as you know, the pepsin works in the stomach. So the pepsin which works in the stomach has a very low pH on which it works. You can see it here. The salivary amylase works around near the neutral pH. And then trypsin, you can see, works around here, an alkaline pH. So you have to understand is that how the different enzymes have different pH and that pH is maintained. Similarly, here you can see optimal pH for stomach enzyme and optimal pH for the intestinal enzyme. So you can be asked a question on that, you can be asked an MCQ on that. And because it says understand that the different proteases present in the stomach and the duodenum work best at different pH levels. Now we've understood that bile contains bile salts. So we've got these bile salts. Then you have a large fat droplet. You have this large fat droplet. So the bile salts emulsify fats. They break this large fat droplet into smaller droplets. And they've coated it with the bile salts, and that has made them into smaller fat droplets. Now, what is going to happen is that the enzyme lipase is going to work on them. So it increases the surface area. You see, it would work on this also. I've explained this in a previous video as well. But now it increases the surface area, so large surface area for the enzyme lipase to work, because the lipase enzyme is going to digest it into fatty acids and glycerol. Now that is essential that we have this very efficient. If they are large fat globules or fat droplets, then it will take longer for the enzyme lipase to work. So the enzyme lipase is going to digest this into fatty acids and glycerol. So in order to facilitate that, we have the bile salts which are going to emulsify the large fat droplets into small fat droplets. So here we have a small surface area to volume ratio. Here we have a large surface area to volume ratio. And now another very beautiful diagram to show you the starch into simple sugars, the proteins into amino acids, and then the bile salts in the lipase, which converts the fats into fatty acids. You can see here now how they've shown you this is the glycerol, so this is the glycerol here, and then you can see the three fatty acids. This is the three fatty acids, and you can see them here. So I remember there was a question on this as well. Now the last topic in this is peristalsis. Now if you look at peristalsis, it's a series of involuntary wave-like muscle contractions which move food along the digestive tract, entire digestive tract, not just the esophagus. So now you see the direction is like this. So now what is happening behind the bolus? The circular muscle is contracting. The longitudinal muscles are relaxing. And the bolus is pushed ahead. Then again as it goes ahead, then here is contracted behind the bolus. And in front of the bolus the muscle is relaxed. So peristalsis is a wave-like muscle contraction. So we've got the two types of muscles, circular muscles and the longitudinal muscles, which contract and relax. So we've got the circular muscles and we've got the longitudinal muscles. Then we see another diagram, same here, section of the esophagus, smooth muscles. Contraction of smooth muscles behind the bolus forces it forward. Waves of muscle contraction move the bolus towards the stomach. Another diagram for you to see to make it. I hope you all these diagrams which I choose makes it all very easy for you to comprehend, because I believe in actually seeing diagrams and then understanding. I don't believe in a lot of theory. I believe in you to be able to see diagrams and make mental pictures of it and mental images of it, that what am I talking about. So the area of contraction would be behind the bolus, because the bolus is moving like this. The bolus is moving in front. So you can see here contracting behind the bolus, and then it is relaxing in front so that it can go ahead. Area of contraction and area of relaxation. So this is what you all have to understand in the thing on peristalsis. This completes chapter 8.2, and the rest of the chapter, which is 8.3, will continue in the third video, which is about absorption and assimilation. Thank you very much for subscribing. Thank you very much for watching. Thank you for all the comments. And thank you once again.